Hello? Okay, that's better. Okay. So first I want to tell you that trying to use the laser pointer and pointing to the screen is just like scoring from the corner in the soccer in the Euro Cup. But luckily we got this mobile microphone, so we'll be able to go down and present from there. The symposium is about revealing fine-scale representations and processing with high-resolution fMRI and MVPA. The uniformity of the mammalian cortex has led to the proposition that there exist elementary cortical units of operation consisting of several hundred or thousand neurons that are repeated within and across cortical areas. Cortical columns and layers of neocortex are prominent examples of such structurally and functionally specialized units. Functional properties and connectivity are similar for neurons within a column, but are known to vary between columns. It can therefore be argued, based on information theory, that the optimal spatial scale for studying the relationship between brain function and behavior is that of cortical columns and layers. The symposium will focus on cutting-edge methods and results of combining high-resolution functional imaging and multivariate pattern analysis for probing fine-scale cortical representations and processing. I would like to invite the first speaker, Merriam from NYU.
Can you hear me? Good? Nope? Better. Okay, this is me. I'm going to be talking about work that I did at NYU with Jeremy Friedman, Helena Wang, and David Heeger. And I'm going to start with the very uncontentious proposition that my slides will not advance. Hello? Tech support? Guys, help. Tech support, I got a problem. Oh, too far. Okay, start again. Okay, understanding the neural basis of perception and action is going to depend on reliable and robust measurements of neural activity at multiple spatial scales, ranging from the scale of individual neurons in networks all the way up to the scale of large-scale cortical networks in the brain. Now, human fMRI is sort of naturally suited for measuring activity at these two spatial scales. So this is the scale of topography within a cortical area, in this case, primary visual cortex, and this is the scale of large networks in the brain. And the question that I'm going to ask today is whether we can use conventional human fMRI to sort of push the boundary and measure these finer scale representations in the human brain. I'm going to start by describing a very old and tried and true method that we use all the time in the lab, and that's retinotopic mapping. In a typical experiment, subjects fixate the center of the screen, and a very salient flickering checkerboard slowly rotates around the screen. And this stimulus evokes a traveling wave of activity in cortex. And we can use standard Fourier analysis methods to characterize the amplitude and the phase of the response in each voxel. And doing this enables us to create color maps like this in primary visual cortex that represent the preferred spatial location for each voxel. So, for example, a green voxel over here responds most strongly when the stimulus is located in that portion of the screen. So this is a retinotopic map in primary visual cortex. And the question is whether we can use fMRI to measure fine scale selectivity for features within this map. And the stimulus feature that I'm going to focus on is orientation. Now, we know a lot about stimulus orientation from decades and decades of neurophysiology. Of course, Hubel and Wiesel discovered that individual neurons in visual cortex are orientation selective. V1 neurons have smooth orientation tuning functions, which means that they respond strongly to their preferred orientation and very weakly to the null orientation or orthogonal orientation. And V1 neurons are arranged in columns and pinwheels where nearby neurons share an orientation preference. And this is an optical imaging map of V1 where each color indicates a different orientation preference. So can we measure this sort of activity with conventional fMRI? Well, typically fMRI voxels are pretty big. Here's a simulated 3 millimeter voxel. Now we routinely measure the brain at 1.5 or 2 millimeters. But still, voxels are much, much bigger than cortical columns. So initially, this doesn't look good. One solution is to use very high resolution fMRI. So we can just decrease the size of our voxels. And a group in Minnesota has done this. They've scanned with very, very small voxels, so 0.25 millimeters on a side, and have shown ocular dominance and orientation selectivity in the human brain. Of course, they had to find subjects with flat calculated sulci because they only had one slice, and the SNR was a real issue, so they had to do a lot of signal averaging, and this is just really, really hard to do. So wouldn't it be nice if we could devise statistical procedures borrowed from machine learning to pull out orientation selectivity in humans using conventional fMRI? So I'm going to talk a little bit about the fMRI decoding paradigm, or MVPA, which was introduced by Haxby several years ago in a science paper, and then picked up on in the field. And if you walk through the halls of this poster session today, there are many, many posters that use this technique to measure selectivity in the brain in many different domains. So the basic idea of this paradigm is you show the subject a bunch of stimuli, you divide the data into two halves, you train a classifier on one half, and then you test the classifier that you've trained on the other half of the data. And this has proven to be an incredibly sensitive method. This works in the case of Haxby's study to decode or classify objects, but it also works to classify orientation. So here I'm showing you a figure from Boynton's review paper that was a commentary on a paper by Kamatani and Tong in Nature Neuroscience about a decade ago. And what they found is that you can train a classifier to decode orientation. The conjecture at the time was that classification works because small biases in the fMRI response arise because voxels sample in a regular underlying columnar architecture. So columns are irregular, voxels are regular. So just by chance, you might sample from slightly more columns that have a preference for 45 degrees, say. And the next voxel over has a slightly different orientation preference for the same reason. And while each bias in every individual voxel might not be significant, if you pool the data using MVPA, you can pull out the orientation that the subject was viewing. So this idea got very popular in the field. These papers have been cited over 1,000 times each. But I'd like to emphasize that this is just a hypothesis. We know orientation decoding works, but we don't really know why. This story is just a hypothesis. So what Jeremy Freeman, David Heeger, and I and others did was set out to test that hypothesis using the retinotopic protocol that I showed you before and what we call an orientation-otopic protocol, where we slowly vary the orientation of the grading as the subject fixated. Retinotopic mapping, subjects fixate in the stimulus location changes, and the orientopic mapping, subjects fixate in the orientation of the grading changes. Just to remind you, this is what a retinotopic map looks like, where color indicates preferred spatial position. And the question is, what does the orientation map look like? If this story is correct, then every voxel should have a different color. Should just look random. And what we are surprised to find is that there is a map for orientation bias, completely unpredicted by the previous physiological literature. So this is not a map of retinotopy, this is a map of orientation preference, although the two maps look very, very similar. Why? Oh, first I want to tell you that this orientation bias map is very small. So typically in our retinotopic mapping experiments, we see a large response, about 2% signal change. The orientation selective response is much smaller, about 10 times smaller. While it's small, it's highly reliable. And we're able to make figures like this summarizing the data, where each voxel is represented by a line. The orientation of the line shows its preferred orientation. The color indicates the signal-to-noise of the measurement for that voxel. And you can see that most lines point inward. So this is an inward-pointing orientation bias, or a radial bias in primary visual cortex. And one question that we found interesting is, what's the neural basis for this orientation bias? If the single-unit physiologist missed this, going one neuron at a time, where is this coming from? One possibility is that it's inherited from the very first stages of visual processing. So there's a very old literature dating back to the 80s by Jeff Shaw, actually, during his graduate work, showing that retinal ganglion cells have an orientation preference. And the orientation preference tends to be towards the retina, towards the center. So there is a radial bias in the retina that might be propagated forward through LGN into V1, and that's what we're picking up on in V1 with fMRI. Another possibility is that there's some impact of the aperture. So we and everyone else in the field places stimuli inside apertures. Either we have a circular aperture on the screen, or the screen itself acts as an aperture. And in a very nice theoretical paper, Carlson in Journal Neuroscience about 2 years ago showed that vignetting a stimulus with an aperture affects the orientation tuning of simulated V1 neurons. We entirely agree with this conjecture, or this demonstration, I should say, but our take is slightly different. So consider this vertical grading here. I'm showing the stimulus in the image domain and in the Fourier domain over here. Those two black dots are the Fourier power of the stimulus. I'm now going to show you two simulated neurons. There's a green neuron and a red neuron. The green neuron has narrow orientation tuning, but broad spatial frequency tuning. I should say narrow orientation, broad spatial frequency. The red neuron, neuron 2 here, has broad orientation tuning and narrow spatial frequency tuning. But critically, both simulated neurons respond strongly to this stimulus. They both like vertical. Now, what happens when we put a vignette on the stimulus? So this is a vertical grading vignetted by a vertical aperture that smears the power of the stimulus in this direction. Now, neuron 1, the green neuron, responds very strongly, but the red neuron doesn't change its response at all. Similarly, if we put a horizontal vignette on this stimulus, we smear power this way. Now the red neuron is responding more strongly, and the green neuron is not modulated. So I've introduced apparent orientation tuning without changing the orientation of the grading. And this may be influencing our results. But I'm going to tell you here, it doesn't matter. It doesn't matter if it's neural, inherited from the retina, or whether it's due to the aperture. The point is, this is a very prominent feature of fMRI measurements of orientation. And the question is whether this coarse scale thing, this big thing, is what's driving decoding, or whether this conjecture by Kamentani and Tong is what's happening. To address this question, we did the following 3 analyses. Now, remember, for every voxel, I have 2 measurements. I've got orientation selectivity and polar angle retinotopy. And I'm going to group the data by the polar angle preference. I'm then going to average the data in bins. So I'm going to take like these 5 voxels here, selected based on their polar angle preference, and average them in the orientation mapping experiment. And if decoding is due to small biases arising from the irregular structure of columns, this averaging procedure should diminish decoding. And in fact, if we average the data in bigger bins, then decoding should get even worse because I'm disrupting the fine-scale columnar architecture. And what we found is that we can decode from these average supervoxels all the way down to 3 bins. So I'm averaging hundreds and hundreds of voxels together. And we still get reliable decoding for orientation. If I average voxels randomly, decoding falls to chance. So this is evidence that the coarse scale orientation bias is sufficient for orientation decoding. We also showed that it was necessary. So here are my two collection of voxels again. What we did is we removed the polar angle component from the orientation measurements. And we found that decoding, which was initially quite high, fell to chance. So the orientation map is both necessary and sufficient for orientation decoding. One more experiment, which I found very convincing, if I do say so myself, was that we moved the slices around. So typically in fMRI experiments, you stabilize the head, you ask the subject not to move, and you, of course, do not move the slice prescription. In this experiment, we stabilized the subject on a bite bar, and on alternating runs, we actually shifted the position of the slice by half a slice width, by 1 millimeter. And we trained the classifier on half the data and tested on the other half, and we found that actually disrupting the mapping between column and voxel had absolutely no impact on decoding accuracy for spiral gratings or radial orientation gratings. So by sampling different cortical tissue, there's no impact on decoding. Interim summary. Coarse scale bias is necessary and sufficient for decoding, and we found no evidence that irregularities in the columnar architecture contribute in any way. All right, maybe that's specific to orientation. Maybe I've just picked one case where it's impossible to see a columnar bias. So what about other stimulus features? I'm now going to turn to MT and motion processing. We know from a beautiful literature on the organization of MT that there are columns that are motion selective. All neurons within a column are selective for the same direction of motion. MT has motion selective columns, V1 does not. So if the columnar conjecture is true, we should see robust decoding of motion direction in MT and weak, if any, decoding in V1. We did an experiment that was analogous to the orientation experiment that I told you about earlier. In this experiment, subjects fixated and viewed a field of dots that moved quite quickly, 5 degrees a second, and the motion direction changed on every TR, every second and a half, completing one cycle in 24 seconds. I'm now going to show you a couple of maps on the brain, and I'm going to color code the map by their preferred orientation preference. There's a lot here. The first thing I want you to notice is there's hardly any motion direction selectivity in MST and MT, the two areas that we expected to see the strongest motion selectivity. But there's robust motion responses in V1. The second thing I want you to notice is that there's a difference between voxels near the fovea and in the periphery. So if you look at the right hemisphere here, near the fovea, there are a bunch of red voxels. Those correspond to motion directions to the left, so away from the fovea. Blue voxels in the periphery corresponding to motion inward, so away from the edge of the aperture. The opposite is true in the left hemisphere. So near the fovea, there's a preference for motion away from the fovea, and in the periphery, there's a preference for motion inward. To make this clearer, we altered the experimental protocol, so there were two patches, one in the left visual field, one in the right visual field, and we changed motion direction independently in the two patches. And what you can see clearest in this inset over here is that there's a clear progression in motion direction away from the edge of the aperture. So what could be causing this? Well, there's a long literature on competitive interactions within V1 where one neuron's response suppresses its neighbors. And we wondered if perhaps there was suppression along the path of motion where the strongest response was associated with the location where the dots appeared, and successive steps along the path of dot motion were smaller and smaller. To test, I'm going to skip this slide. To test this hypothesis, we did the following experiment. We confined the dots to two rectangular apertures, and we alternated inward and outward motion in blocks of time. So this allowed us to measure the response to a block of outward motion, followed by a block of inward motion. We then plotted the amplitude of the response in V1, shown in blue, and MT, shown in red. I don't have the labels here, so you'll have to take my word for it. And what you can see for fovea outward motion, there's a strong response, Fovea outward is down, fovea inward is up. So away from the fovea, the strongest response is near the fovea. And out here, the strongest response is inward, okay? And at intermediate locations, there's an intermediate response. So that's consistent with a gradual suppression of motion along the path of the moving dots. So these are two different cortical mechanisms. I've described a neural and an aperture mechanism to explain the orientation map, and a suppression along the path of motion explanation to describe the motion map in V1. Neither depend on a columnar conjecture. So to conclude, fMRI decoding is a powerful tool that can detect interesting coarse-scale structure and highly complex multivariate data. Coarse-scale biases for orientation and motion provide insights into cortical computation. And finally, this is a vision talk. I'm a vision neuroscientist, but I think by studying vision and gratings and moving dots, we can learn a lot about the nature of the signal that we're measuring as neuroimagers, and I think these principles will extend nicely to studying other parts of the brain. Thank you.
We have time for one question.
You.
Yeah, thank you. This was great. So I was wondering how to interpret your findings. I feel like there could be three options. One option is fMRI is just too coarse-grained to pick up orientation columns. Option two is orientation selectivity is a network property and not like a property of individual neurons. And option three would be it has something to do with like population receptive fields, perhaps.
I don't know if those are mutually exclusive. I'd have to think about it some more. So the first option, the fMRI is just too coarse to pick up on columns. I think that's true for conventional fMRI, but of course the conventions are changing, and my hope is in a few years measuring columns is going to be routine. And I think ultimately we'll want to measure columns and maps and networks all in the same session simultaneously. I think it's certainly true that these computations that I've talked about today are network properties. I think we don't understand a lot about the physiology for how these computations arise. So, I've got collaborations now with physiologists who are doing the same experiments with arrays, and I think those are going to be informative. Thank you.
Thanks.
Thank you very much. I'd like to invite the technical support here, please.
The next talk will be by Amir Shmuel from McGill University in Montreal. And the title of his talk will be Evaluating Contributions of Fine-Scale Irregularities, Low-Frequency Organizations, Macroscopic Blood Vessels, and Spatiotemporal Responses to fMRI-Based Orientation Decoding.
Can you hear me in the back? Can you hear me in the back? Doesn't work. Mechanisms of fMRI-based decoding of information conveyed by cortical columns. So we know now for about 11 years that fMRI at a resolution of 3 by 3 by 3 millimeter can decode the orientation of grating stimuli. This is puzzling because the voxel of 3 millimeter is coarse relative to the fine scale organization of orientation. There have been 5 classes of mechanism proposed to account for this phenomenon. Aliasing, contributions from fine-scale irregularities, low spatial frequencies arising either from large-scale organizations or from edge effects, selective responses from draining veins, or complex spatiotemporal filtering by fMRI voxels. We aim to unravel the mechanisms underlying hemodynamic response-based decoding of oriented grating stimuli using coarse voxel samples. We are interested in this question because unraveling the mechanisms underlying decoding of fine-scale organization will enable informed search for such organizations in relatively unknown cortical areas. We started this journey by a model put together by Denis Kaimov 5 years ago. It's a mathematical model, and we tested two proposed mechanisms, one of aliasing and the other of irregularities. The model starts by white Gaussian noise and by filtering with anisotropic filter in order to obtain columns-like patterns similar to ocular dominance columns in humans. We then simulated neuronal response to either stimulation of the left eye or the right eye, convolved with the bold hemodynamic point spread function, and simulated K-space sampling. With the voxels, we analyzed mechanism of fMRI-based decoding. So the first question, is there aliasing in fMRI? Well, we tend to think about voxels in the image space as if they are rectangular. However, because of the way K-space samples fMRI data, voxels in the image space, in fact, are sync-like function. And here we see the representation of the sampling in the K-space, where a maximal frequency is defined in blue. That's the definition of the maximal frequency that we can get with that specific acquisition. And superimposed is the power spectra of ocular dominance columns. We see that the fMRI sampling with 3 by 3 by 3 millimeter voxels can be sensitive only to the low tail of the frequency spectrum, very low frequencies in the ocular dominance map. If the voxel was a rectangle-like in the image domain, then aliasing could be possible. But with K-space sampling, aliasing is not possible. We concluded other questions that we addressed with the model, and we addressed questions about aliasing and irregularities by ruling out contributions of aliasing and concluding that contributions from fine-scale irregularities are small, if any. We then proceeded to experiments in cat area 17. The reason we use animals here is that we can get to very high resolution, 156 micron per voxel, and also because we can use a myon, which is a contrast agent that enhances contrast to noise ratio, but most importantly, we were interested in isolating contributions from irregularities and low spatial frequencies. In order to isolate them, we wanted to exclude contributions from macroscopic vessels or from spatiotemporal responses. Well, when pursuing fMRI with myon, the T2 star of myon in macroscopic vessels is extremely short. Virtually all our responses are from the tissue. In addition, we use a phase encoding paradigm in which the gratings, while moving back and forth, also rotated slowly and therefore changed the orientation very slowly. This creates a situation in which the cerebral blood volume in macroscopic vessels is approximately constant. Again, that's another reason why we don't get here contributions from macroscopic vessels or propagation of spatiotemporal responses. So here is a voxel, one voxels in cat area 17. We see here the phase encoding time course of this voxel and how it looks in the frequency domain. This is the frequency of the stimulation. From the phase of the stimulation, we obtained very fine scale orientation maps in a cat area 17. Now, in order to further analyze the data, we computed the contrast, simply differential responses or differential analysis of responses to one orientation minus an orthogonal orientation, the noise, the variability of a trial, and the contrast to noise ratio. Then we pursued exactly the same analysis, frequency, by first filtering the responses around a narrow band or in a narrow band of frequencies around the center frequency for all frequencies. This way, we got the contrast to noise ratio as a function of cortical frequency. This is data from 2 different animals, and we can see that the peak of the Organization for Orientation Maps is around 1 cycle per millimeter, and a Gaussian fits the spectrum around this peak very well. This is from a second animal. Note, however, that the distribution is bimodal. We see here also low frequencies. Now, it is, because the distribution is bimodal here, it is logical to define the irregularities domain around the main peak of the organization and to lower frequencies up to the trough where the low frequencies start. So this way, we defined irregularities and low frequencies domains in cat area 17. Now, we need to match the voxel size between humans, 3 by 3 by 3 millimeters, and cat. We did it by comparing the peak power of the Organization for Orientation. In humans, we expect, and we partially know from published studies, that the orientation cycle is approximately, 1 orientation cycle is approximately 2 millimeters. And Kamitani and Tong use 3 millimeter voxels. Therefore, with 1 millimeter per cycle in cat area 17, the voxels matched in size to humans is 1.5 millimeter. And what we can see here is that the voxels that matches in size does not or cannot capture frequencies in the irregularities domain, only in the low frequencies domain. In order to further verify it, we computed decoding rate, support vector machine decoding, following low-pass filtering and binning at multi-scale filtering and binning, including voxels of 1.5 millimeters that are matched in size to 3 millimeters in humans. We then computed the decoding rate following low-pass filtering, where the vertical axis here represents the filter cutoff of the low-pass filtering, and the horizontal axis represents the bin size. Not surprisingly, we see here elevated decoding rate wherever the low-pass filtering included frequencies of the main frequency of the organization, which is 1 cycle per millimeter, or voxels that are smaller than 0.5 millimeters that can capture these frequencies. But we are interested in the voxels that are matched in size, and this voxel is 1.5 millimeter. So we can sample from this matrix of decoding rate all the decoding rates with the voxels match in size and present it in this plot here as a function of filter cutoff. So the voxels matched in size, the decoding rate with the voxels matched in size is presented by the dark plot as a function of a cutoff frequency of the low-pass filter. What we can see is that the decoding rate remains constant from 0.33 cycles per millimeter to higher frequencies included after the low-pass filtering. In other words, the size match voxels captures only low frequencies. We were also interested in what would be the largest voxel that can still captures the irregularities domain. This turns out to be 0.75 in cat area 17, which is matched in size to 1.5 millimeter in humans. So we concluded this part of the study by saying that using Myon-based fMRI, fine-scale irregularities do not contribute to decoding of orientation stimuli. Since the spatial specificity of CBV fMRI is higher, is better than that of bold fMRI, we can conclude that irregularities do not contribute to bold fMRI-based decoding of orientation stimuli. Furthermore, contribution from macroscopic blood vessels and from spatiotemporal propagation of fMRI responses are not necessary for decoding orientation stimuli, because with all the parameters that we used, we were able to decode the orientation. However, our signal did not include any contribution from macroscopic vessels. So we started by being interested in checking for contributions of irregularities and low spatial frequencies, and we concluded that in cat area 17, irregularities do not contribute to decoding, low spatial frequencies do contribute, macroscopic vessels and spatiotemporal responses are not necessary for successful decoding. We then were interested whether this result will generalize in other areas. Do these results depend on the cortical area? In order to address this question, we used optical imaging in cat area 18. We used optical imaging so that we can also test contributions directly from macroscopic vessels. So here we tested for contributions of irregularities, low spatial frequencies, and macroscopic vessels. The principle of optical imaging is that when we remove the scalp in an animal model, and we image directly from the cerebral cortex under two wavelengths of illumination. The principle of the measurement is based on different absorption of the oxy and oxyhemoglobin as a function of wavelengths of illumination. Based on data from two wavelengths, we can compute changes in the oxyhemoglobin, which approximates bold fMRI, and changes in total hemoglobin, which approximate cerebral blood volume fMRI. We also delineated cortical vessels, or pial vessels, and identified arteries and veins in order to check contributions of draining veins to decoding of orientation. Here is the contrast to noise, the noise and the contrast to noise ratio obtained from deoxyhemoglobin and cerebral blood volume or total hemoglobin responses in response to orientation, stimulation with orientation. Similarly to the study in cat area 17, we repeated this analysis frequency by frequency and obtained the spectrum of contrast to noise ratio as a function of cortical frequency. The peak of the organization in cat area 18 is 0.84 cycles per millimeter, so the columns are wider. Also note that unlike in cat area 17, the distribution here is not bimodal. We can see a Gaussian-like spectrum with no elevated power in low frequencies. So we can model the responses in area 18 as belonging only to irregularities, including responses at the very low frequencies that are still in the domain of irregularities because there's no low spatial frequency organization. Now, the size match voxel is 1.8 millimeter, and it captures frequencies up to 0.28 cycles per millimeter in cat area 18. Size match voxels in cat area 18 captures frequencies at the tail end of the Gaussian-like contrast to noise ratio curve. We then went on to compute directly decoding with support vector machine, this time from gray matter region, separately from large vessel regions, and from gray matter and large vessels combined. We can see that the decoding rate from gray matter region is very similar to the decoding rate obtained from gray matter and large vessels, and the decoding rate from large vessels is smaller than both. We further computed a statistical significance of this phenomenon. So to the left, we see a statistical testing of contributions or decoding rate of gray matter and large vessels minus decoding rate from gray matter, and we can see that nothing is statistically significant. Virtually, these responses or these decoding rates are the same. However, when we subtract large vessel contribution, large vessels decoding rate from decoding rate obtained from gray matter and large vessels, we do get statistically significant differences. This suggests that the contributions from large vessels, although we can decode in a statistically significant manner from large vessels, the contributions of large vessels is redundant when signals from gray matter are included. We repeated this computation now for cerebral blood volume, for total hemoglobin, and we get a very similar pattern where decoding rate from gray matter is similar to decoding rate from gray matter and large vessels and much higher than large vessels. and that the contributions or the decoding rate from large vessels is redundant relative to gray matter. This slide quantifies it. So we see here first in gray that following low-pass filtering with the voxels matched in size to 3 millimeters in humans, following a low-pass filtering with including frequencies higher than 0.28 cycle per millimeter, the decoding rate in gray does not increase, meaning there is some limit and that's it. And we can also see that the decoding rate from gray matter and large vessel in green is higher than decoding rate from large vessels and approximately similar to decoding rate from gray matter in gray. And lastly, decoding rate with cerebral blood volume here is higher than decoding rate with bold right here. So we can conclude from the study in Cat Area 18 that irregularities from the low-frequency tail of the spectrum in the organization of Area 18 underlie orientation decoding, while responses from macroscopic vessels are orientation selective, the contributions to decoding are redundant. Overall conclusions. MVPA is a powerful technique, but there is no magic. With 3 millimeter voxels, you get what you can get with 3 millimeter voxels. Contributions from irregularities depend on the features of the organization, such as the peak frequency, the width of the power around the main frequency, and the voxel size. Whether irregularities or large-scale organizations contribute to decoding depends on features of the area-specific columnar and low-frequency organization. While responses from macroscopic vessels can be orientation selective, their contributions to decoding are redundant. So how do we make sense out of that, and what do we do practically? We can visit a poster by Denis Chaimov that quantifies or optimizes or suggests how to optimize contrast to noise ratio and detection of voxel-wide responses in fMRI of cortical columns, poster 1659. He also optimizes detection, decoding, and reconstruction of cortical columns from fMRI pattern responses, poster 2172. And lastly, he recently fitted the model that I presented at the very beginning to ocular dominance columns data that I obtained with Issa Yacoub previously at the University of Minnesota in order to quantify the spatiotemporal specificity, the spatial specificity of the functional MRI blood oxygenation response relative to neuronal activity in a differential analysis setting. And lastly, we present the spatial specificity of Pial vessels as a function of their diameter with implications for optimizing fMRI of cortical columns. I would like to thank my colleagues, Denis Chaimov, Zeshan Yao, Javid Shaikh, Issa Yacoub, Camille Ugubil, Sengi Kim, Mitsushiro Fukuda, Chang Hong Moon, and my funding agency, NSERC, and you for your attention.
We have 1 minute for a question.
Over here. Amir?
Yeah.
I don't know. I've been doing fMRI for 26 years. I think there's a lot of magic. But beautiful work.
Thank you.
I think maybe you alluded to it in one of the posters you flipped through. Did you consider the point spread function as well of the acquisition? Because that's going to broaden out your sync function, of course. And probably, I don't know how that necessarily changes your MVPA, but can you comment on that?
Right. So in the model, of course, we accounted for the point spread function of the bold response. In the experimental data, we didn't need to account for it because it is already present in the data in terms of the point spread function. So, of course, we accounted for it. And as I mentioned, we have now a poster that quantifies the point spread function in a differential analysis setting. Our previous paper looked at the point spread function in a single condition setting, and there is a difference. You know, if you think about everything as linear systems, then there should not be a difference. But there is a difference because fMRI, especially with gradient echo and with spin echo fMRI, has contributions of distance. So the point spread function in differential setting is different than the point spread function of single condition setting.
Yeah, so that's probably typically going to be probably a millimeter at 7T or thereabouts.
Yeah, that's correct. So with gradient echo, we get a point spread function which is about 1 millimeter. With spin echo, we get about 0.8 millimeter. Thank you. The next speaker will be Lars Muckli from the University of Glasgow, who will talk about layer-specific brain imaging of top-down internal models induced by visual and auditory context.
Thank you, Amir. Thank you. Good afternoon. I will talk today. I will approach the topic of fine scale with the focus on laminar fMRI rather than columnar fMRI. And I will do this by talking about how the internal models that we create, for example, by mind wandering or imagery, can be read out in primary visual cortex. Primary visual cortex area V1 is interconnected with many areas. And those areas it is feeding to, it receives feedback. It's reciprocally connected to many clever areas, and they are contributing in their feedback signal some clever calculations about what they are processing. It's highly interconnected, think of it like the UK before the Brexit. If you look at primary visual cortex, at the laminar structure, there's a very interesting pattern that emerges. The feed-forward connection reaches into the layer 4 and 5, and it does so to neighboring areas as well. So the feed-forward stream feeds into the layer 4 and so on. The feedback stream feeds to the superficial layers. The superficial layers 1, 2, and 3, and also the more deep layers, layer 6, as shown here. And there's an interesting pattern that was shown by Markov and Kennedy, reviewing 25 years of monkey anatomy, showing that from neighboring areas, proximal areas in the hierarchy, like if you take V2, then from V3, feedback would predominantly go to the superficial layers as well as the deep layers. Whereas if you take more remote areas, the feedback tends to target more the deep layers. There's also a neuronal mechanism that has been proposed by Matthew Larkham that identified layer 5 pyramidal cells that have the property to read out the dendritic feedback signal in the superficial layers and combine that with dendritic input at the proximal integration zone of this pyramidal cells. And if the information fits between the dendritic feedback in the proximal area to the distal and the proximal fit together, then these cells engage in bursting property, so they can integrate this kind of information. If you stimulate a column, for example with a checkerboard, then you have activity usually throughout the whole column, through all the different layers, but you receive feedback because the higher areas like V4, in this example from Charles Gilbert's lab, you see the receptive field properties of V1 in red and the receptive field properties of V4 in green, and they're much larger. And they can feed back information, for example, from a collinear line pattern that you see in the top, and that feeds back to area V1, and this feedback spreads out, if you like, to the superficial layers, and there are some cells in V1 that you can record that have this property to integrate this feedback and respond differently depending on this remote receptive field so outside of the receptive field effect. Now that's the idea that we used in our fMRI experiment, in which we used high-resolution 7T imaging with 0.8 millimeter voxel resolution, and we did retinotopic mapping to map out precisely the region of the occlusion in the right lower corner of the image, which we occluded. This area is just responding, this you can see in orange, is just responding to the target area, but we're not interested in the boundary area. Then we took the outer PL surface and made depth level, six cortical levels, if you like, from the outer PL surface to the gray-white meta boundary, equally spaced. And those layers, we then used the voxels in these layers to do our further analysis. The stimulation analysis is in two ways. One, the red one shows we actually stimulate those voxels. You see the receptive field, the population receptive field symbolized with these small circles, showing now in this regime you have voxels that are actually activated. And in the green paradigm, these voxels are just looking to a white occluder, and they are not activated, they are just baseline activity. So we don't use activation analysis, but multivoxel pattern analysis, so a way of information encoding. We do that by training the classifiers in 3 runs and then testing it in the 4th run. And by doing this, we can see in the 4 individual subjects that if the voxels are stimulated in all of the layers, so the deep layers shown to the left and the superficial layers shown to the right, there's enough information to do MVPA decoding. So that's a sanity check, which is good. And on the lower, and the green lines, the 4 green lines are individual subjects from the same voxels, but now during those conditions in which they are not really stimulated, so they are baseline activity. And in most depth layers, especially in the deep and the middle one, you have no information to do the decoding, except in the superficial layers shown towards the right, there's enough information to decode the surround scene. And this information we propose is coming from higher visual areas like V4, having information about the scene, feeding back to V1. This is the average about the group. So we can conclude that in the superficial layers of area V1, we have feedback information about the spatial context shown with MVPA analysis. We used gradient echo EPI imaging, and there's a little bit of a confound, so let's look at this. So in gradient echo imaging, it's often observed that in the outer surfaces, seen towards the right, in the superficial layers, you have higher bold amplitude. Those are our 4 subjects, and they always have higher bold amplitude towards the right. Those are the superficial layers, relative to the left, which are the deep layers. However, in the occluded condition, that doesn't really matter because they are not stimulated. These voxels are in their baseline activity, and they don't show any of these bias, except maybe of one subject. So the information space you see in the bottom shows that on the right, the superficial layers have this information despite a flat bold distribution across the different layers. So we can conclude that the bold amplitude and information encoding is unrelated. We further confirmed this conclusion by using grade 3D bold imaging, which, as you can see, has a flat property across the different cortical depth levels. The amplitude is always the same, even for the stimulated condition, but also during the non-stimulated condition of the occlusion paradigm. And you see in the 2 examples at the bottom that feed-forward can be encoded, the information is present, and for the feedback condition shown in the green, the information is only in the superficial layers. We use this paradigm now for a parametric design. This is 3T data analysis of many subjects. Each of those dots is one individual subject. We trained our classifier between 2 images. One is the car and the other ones are people. They are contrast matched. And so once the classifier has enough information to brain read between these 2 conditions, we challenged the classifier by moving the stimuli for 2 or 4 or more visual degrees angle, and then tested the classifier against without further training with the new stimuli. And as you can see, the information drops after 4 degrees. So it also drops between 0 and 2. So there's some fine-grained structure, some fine-grained information in the feedback, but there's also a plateau up to 4 degrees in which there's still information remaining, which makes sense if the feedback from before is more coarse. So we use the same paradigm now with 7T brain imaging, again with 0.8 millimeter resolution. And you can see in the pink lines are those shifts that are 2 degrees. The other lines are further larger shifts. So we can conclude that only 2 degrees are significant, indicated by the dots on the lines. Those are permutation-tested significances. And you can see they're only towards the right, which means the superficial surfaces. So it's only in the superficial surfaces, and it sustains shifts of 2 degrees. Now let me focus a little bit on the scale of the information pattern. Here we use a different technique based on GLMs to see which are potential voxels that have some property that can be contributing to the information scale. And those are the target regions in the top line and the midline. It's a very high threshold, and it's a lower threshold underneath. And so you see the color code, the rainbow code that I've been using. Outers are superficial layers are red, and there are more of those voxels. They don't form a very clear pattern, and more details are in the paper. However, one thing that we did is if we now jitter between one run and another, the whole brain by one voxel or 2 voxels or 3 voxels, and then test the classifier in a shifted version of the brain, we can learn something about the spatial resolution of the underlying information pattern. And that's highlighted here on the left for the feed-forward information, and that goes throughout the layers, in all of those layers, and on the right it's for the occluded condition where we only have information in the most outer layers, shown in the purple line at the bottom right. And we can see that it's a kind of a shallow drop-off, and that shows that there's a bit of a less precise feedback signal, if you like. And here are shown the first derivatives, and it shows the difference between the feed-forward and the feedback. The feed-forward is more precise than the feedback signal. Now let's turn to the second paradigm that we have used. We've used auditory feedback to V1. So we blindfolded our subjects, and now we can make sure that there's no feed-forward input in primary visual cortex. And we did 2 paradigms, one with presenting auditory scenes, and the other one with having the subjects engaging in mental imagery of auditory scenes. So to give you an idea how that works, it's total dark, you're blindfolded. And this would be one auditory scene, which we call forest or bird song, if you like. And another condition would be a cocktail party effect or as traffic. Now, early visual cortex, V1, V2, and V3, usually engages in negative bold during listening to those scenes, but for the different auditory scenes, we have the same amount of amplitude change. If you look, however, at the information space, you can see that the early visual cortex has a little bit of information about these different auditory scenes, as well as the auditory cortex, and we also use the control area motor cortex, which doesn't have this kind of information. If we focus into early visual cortex, we can separate V1, V2, and V3, and this signal seems to be coming from V2 and V3. At least we can show that there is some information. The second paradigm is we had the subjects in every other run, we had the subjects in the scanner, and instead of presenting these auditory scenes, we just gave them a cue.
Traffic.
And then the subjects engaged for 12 seconds into imagining the sounds that they have heard before until they hear a beep and they stopped. And now taking the activity of this block of imagery, you can read out the 3 different scenes, again, trained on 3 runs, testing on the 4th run, cross-validation, MVPA analysis, that the auditory cortex knows about the auditory scenes that were imagined, and so does the visual cortex. And if we look into early visual cortex, it seems that it's mainly V1 contributing to this. So now we take these 2 paradigms into 7T environment and look at the layer-specific signature of this feedback signal. So here I highlight 3 of the 6 layers that we separated in our subjects. And now we look at the auditory paradigm, subjects just listening to these 3 different conditions. And this is one subject. And you can see now to the left that in the deep layers of early visual cortex, there's significant permutation-tested response, and also some in the superficial layer. The second subject only has in the deep layer, and the third subject is a little bit more undecided. Now let's move to the second paradigm. That's the imagery paradigm. So now no auditory stimulation, but just the instruction to imagine the whole scene. And there you have information in the superficial layers of 2 subjects, and the third one has it in the deep layers. So how can we reduce the individual variance? We have actually measured 8 subjects so far. We had to get rid of 4 because their head movements, they were unexperienced. We pushed too far. And we got another 4 subjects in that are highly trained to reduce movements. And we spend a lot of time on optimizing the alignment procedures. Here you see a correlation between the 10 runs. 5 of those runs, they're always interleaved, are with auditory stimulation, and 5 of them are with the imagery. But you see that run 9 and 10 is slightly less correlated to one another than the other runs. So this is something that needs to be optimized. Also, with head movements between runs, there comes a different susceptibility artifacts. And so it's important to optimize the reducing the distortions that could be caused by this, and we use a top-up procedure to do that. Other ways to look out for, and why the data analysis is so time-consuming, is to reduce variance by the alignment between anatomy and the EPI images. We optimize the reconstruction, the segmentation on the EPI images, and then construct layer reconstructions piece after piece and piece them together to be really sure that the layers are consistent, which seems to be important. So, conclusion from those paradigms, first of all, there's cortical feedback to the superficial layers. If the spatial contextual feedback, so if the scene is feeding back to the occluded part of the scene, the details of this are published in a paper in Current Biology last year, and you can see that the information is in the superficial layers. That's the first paradigm. And the second paradigm is more work in progress. It shows that in the auditory scene processing, mainly we see mainly information in the deep layers. This is an average of first 4 subjects, actually. And discussion, I want to highlight a study from Peter Koch in Floris de Lange's lab that has looked at a modal completion paradigm, which is related to modal completion is related to our amodal completion. So the difference between these 2 paradigms is that we just occlude, but we don't have an illusion, whereas he is actually inducing an illusion, and he finds different results. So the difference modal and amodal. So in their analysis, they see in the deep layers activity related to the contours of an Cannister square. Now that is related to actually activation analysis, whereas we looked into information analysis. But you see that there's great variance between the individual subjects. And I want to highlight also that it's consistent with what I highlighted at the beginning with the neuronal structure of apical amplification with Matthew Larcombe showing that there's a readout in the top layers saying that in our amodal completion we have something like candidate hypothesis predictions that are lingering in the top superficial layers, and only if they are read out by these layer 5 pyramidal cells in the deep layers, where you then also get activity, you get maybe the illusion related to the illusory contours, and so that would explain why he finds it in the activity pattern and also in the deep layers. And with this, I want to thank my lab, especially Matthew Bennett, who has worked for the data that I've presented here, is now highly trained in the layer-specific analysis. If you want more discussion, you can come to the posters. And Andrew Morgan presented yesterday, still his posters up. Matthew Bennett, that's an extension of my talk. Angus Patton also works on auditory and visual integration. And thank you for your attention.
We need to proceed. I would like to ask the technical support to approach the podium. Thank you. The next speaker is Kriegeskorte from Cambridge, United Kingdom. He'll be talking about testing deep neural network models of cortical processing with fMRI.
All right. I want to talk about testing brain computational models with fMRI, despite the fact that fMRI has rather large voxels, as we've seen in the previous presentations, and measures neuronal activity through the hemodynamics, so gives us a very indirect and somewhat coarse reflection of neuronal activity patterns. So there's this canyon between cognitive theory and BOLD, and we've approached this for a long time by building a kind of bridge across this canyon that I call cognitive neuroscience, where we try to directly relate essentially the boxes of cognitive psychology to regional responses in fMRI data. And this was somewhat compelling. This made sense in a way because both cognitive theory and BOLD operate at a macro level. BOLD is a macro scale measure of brain activity, and cognitive theory set out to kind of define the architecture of cognition in very broad strokes. But of course, we're aware that we really want a more detailed understanding of computational mechanisms, and in order to achieve that, we need to cross this canyon of neuronal detail, and that's a very big challenge. And I think we're all, as a field, struggling with how to go this difficult route and understand computation in the brain in more detail on the one hand, and then also draw more detailed relationships to these increasingly rich data that we can get with brain imaging methods in humans, including high-resolution fMRI. So I want to talk about three questions today. The first is, what does it mean to understand how the brain works? The second is, how can we test brain computational models that perform real-world tasks with fMRI? I think this is an important direction that we have to move in. We need models that are not just descriptive models, not just models that describe our data or allow us to answer questions, but models that actually perform the task. So ultimately, they'll have to be AI models that actually perform feats of intelligence. And then thirdly, I want to talk about how we can account for the way that fMRI reflects fine-grained neuronal activity to infer computational mechanisms. So first, what does it mean to understand how the brain works? To me, it means that we explain neuronal activity patterns, explain behavioral data, ideally in humans, using fully explicit computational models that are neurobiologically plausible and that perform these real-world complex cognitive tasks. And this historically has involved a number of disciplines, but three key disciplines are cognitive science, computational neuroscience, and artificial intelligence. And artificial intelligence is really key to this because obviously a model that really explains brain computations has to be ultimately, if it engages the real world and complex cognition, an AI system. However, beyond other AI systems, it's also supposed to check these other boxes and explain our brain data and our behavioral data. So how can we test these brain computational models that perform real-world tasks with fMRI? In my lab, we take the approach of representational similarity analysis, where we present a number of stimuli to biological brains, humans and monkeys, and to the models. And for each of these stimuli, we get an activity pattern. And then because it's difficult to relate these activity patterns in the models, which can have hundreds of thousands of units, to the responses that we measure in the brains of our subjects, we abstract from this level of the activity patterns and describe the representational space with a particular multivariate summary statistic, which is the representational dissimilarity matrix. So this is a matrix indexed by the stimuli in both dimensions, where you can look up for each pair of stimuli how similar they are in the representational space of a brain area. And because these matrices are indexed by the stimuli, they're very easy to compare between models and brains. So here's an example of the human and monkey inferior temporal representation for 92 different object images that you see very small there along the sides. So this was a data set that we've struggled with for a very long time to explain it. It shows in blue there small dissimilarities and clusters among animates and inanimates, and within the animates of faces. And the matrix for human IT was derived with fMRI voxels, and the monkey matrix was derived with cell recordings. So this is a way of looking in the same framework at data acquired with different modalities of brain activity measurement. So, but the question, of course, is, can we explain this representation with a computational model? So I'm not going to go into the details of these studies, but I want to use them as an example to make some more general points. So when we first started on this journey, we asked whether computer vision representations could explain this representation. So here I'm going to show you the accuracy of the human IT dissimilarity matrix prediction using a number of different computer vision models. So let's start with just one model, the GIST model. So when we tried the GIST model, which is a particular set of computer vision features computed from images, it explained a highly significant amount of variance. So this would suggest that we could write a paper about this and argue that GIST is a somewhat promising model of the IT representation. However, it's very important to look not just at one model, but at many different models, because when we do that, we see that almost all the models we tested explained a highly significant amount of variance. So that means these are encoding models, so they explain variance in the representation by modeling the representation as a function of the stimuli, but you could also invert them, use them for decoding, and then you'd also get significant decoding for almost all of these models. So some of the measures that we're used to looking at, such as decoding and encoding explained variance and whether that's significant, are actually not very useful for learning something about the computational mechanism, because many different mechanisms will explain significant variance. The more important question to ask is whether any of the models fully explain our data. And you see this gray area, here's the noise ceiling. And from looking at this, we see that none of these models actually comes anywhere close to the noise ceiling. So this means that we can reject all of these models based on our data. Our data, despite being limited and noisy, are good enough to reject all of these computational mechanisms. So that's very useful. There we've really learned something about computations. The model that we've been most successful with has been a deep supervised convolutional network. So here again, I'm going to show you the accuracy of the human IT dissimilarity matrix prediction. And layer one of the model explains significant variance, but a very small amount of variance. And as we go up the layers, we get closer and closer to this noise ceiling. And with the final representations, we get into the noise ceiling and, in a sense, fully explain our data. So this doesn't mean that this model is correct. Of course, this model, we already know that in many ways this model falls short as a model for the brain computational mechanism of object recognition. However, it's the most promising model we currently have, and our data are not good enough to reject this model at the moment. So all of these representations are significant, and this highlights the point that I've made before, that just asking for significance is not a very interesting questions from a computational perspective. What's really more important and what gives us hints to how the brain works is the question of whether the model can fully explain the representations that we measure. So there's an emerging body of work using deep neural networks to explain perception, and we're very excited about this. Some major protagonists of this are Dan Yammans and Jim DiCarlo, Charles Cadieux, and also Marcel van Gerven's great lab in Nijmegen. And there are a number of reviews that came out last year and this year that give good introductions to this exciting new direction. I want to highlight one particular study that I saw yesterday at OHBM. There was also a poster, unfortunately, yesterday, so it might be too late to see it. It's showing, among many other things, that a deep neural net model processing a natural movie can predict ventral stream fMRI time courses quite precisely. So here's an example of the FFA, where the model predicts the time course of the FFA response during the viewing of this movie with amazing precision. And I think this really takes this literature to a new level. So this gets me to the third question: how can we account for the way fMRI reflects fine-grained neuronal activity to infer computational mechanisms? So we had these two matrices, but one of them was measured with these coarse fMRI voxels, the other with cell recording. So we might ask, well, shouldn't the appearance of this matrix be influenced by the way it's been measured, right? Why do these look so similar? So here's a possible explanation for why they might look similar. Random projections preserve geometry. If you have p points in an n-dimensional space, and think of this space as the neuronal population response space, if you project them down to a smaller number of m dimensions with random projections, so projecting onto random axes in the space, and you could think of these as ideal measurement channels, then if you have m larger than O of log p random projections, you'll get, with high probability, only a small distortion of the distances that you measure in this lower dimensional space. And this is independent of the embedding dimensionality n. This is the Johnson and Lindenstrauss lemma. And it might explain why even after subsampling and averaging in fMRI voxels, there's some similarity still between the monkey IT matrix measured with cells and the human IT matrix measured with fMRI voxels. So this was very encouraging. However, voxels, of course, are not really random projections, right? They locally average, and they will attenuate the contrast very strongly among neurons that are within the same voxels and less strongly between neurons that are far apart in the representation. So one direction that's important to us is inferring the data-generating computational model under uncertainty about how fMRI voxels downsample neuronal activity. So we have the brain representation. It's sampled with fMRI voxels, and this gives us our representational dissimilarity matrix. And in modeling this, we'd like to model the same process. We'd like to have a model that predicts the spatial structure of the representation. Then we want to simulate how fMRI voxels sample this, and then predict what the RDM should look like given the parameters of this measurement model. And then we can do inference by comparing these. So what does this look like? Here's a simulation trying to address what happens when coarse fMRI voxels sample a fine-grained neuronal population code. So this is RDM space visualized with multidimensional scaling. And this is the color code I'm going to use. So different colors indicate different layers, different representations in this deep neural network model, convolutional layers 1 to 5. And the size of these circles indicates the size of the local averaging range. So you could think of them as small voxels or large voxels. Really, in an fMRI experiment, we know what the size of the voxels was, but we might not know the cortical magnification factor and how much the local vasculature averages. So the averaging kernel size is an unknown parameter that we put a prior over. So when we start with the very small averaging kernels and then sample several times with simulated voxels with these small averaging kernels, we get a number of RDMs, and it's very interesting to see that they're all extremely similar. So these are all independently drawn samples from this population, but the RDMs we get are extremely similar, and this is because of the Johnson-Linton-Strauss lemma. This is very useful to us because it means we don't care what channels of the normal code the voxels sample, each voxel samples precisely. We only care about the summary statistics that describe the representational geometry. So when we make the voxels larger and larger, we get very different RDMs. So this is also a very important observation, which highlights the fact that we cannot ignore the way that voxels sample neuronal activity, because it does affect our summary statistic, the RDM. So here's, I started with convolutional layer 5. Here's the same for convolutional layer 4 and for convolutional layer 3. And here's 2 and 1. So we get these separate subregions in RDM space corresponding to the different brain computational models, and each of them is elongated along dimensions that reflect the parameters of the measurement. So in this case, that's just the local averaging range. So you have these sort of two effects determining these RDMs. And now, of course, if we just ignored the measurement and then we get some RDM estimated from fMRI and infer that it's probably generated by the closest brain computational model predicted RDM, we might make a mistake, right, as in this example where the closest RDM predicted by the brain computational model without any simulation of averaging is the wrong one. However, if we simulate these measurements, we have a chance of inferring the right brain computational mechanism. So when we look at a number of ground truth simulations where we simulated voxels with some averaging range, but then we pretend we don't know what the parameter of the measurement model was, and then we consider a number of candidate models, and for each of them we have different settings of the measurement parameters, then for a given simulated region, we get large variations, as you see as you look along this row. There's blue and there's red, so the measurement model has a huge effect on the RDM. However, when we choose the RDM that has the highest correlation, it's generated by the correct brain computational model, in this case layer 4. So we can do this more formally by simulating thousands of these RDMs from each of the brain computational models for different draws from the priors over the parameters of the measurement model, and then infer the posterior, the probability of each brain computational model given the data. And when we do this for our for simulated subjects here, we see that this tends to work to some extent. So here's the group results, and for each of the data generating model, this analysis correctly infers which model generated the data. So the conclusion from this is that our simulation suggests that the data generating brain computational model can be accurately inferred despite the local averaging in fMRI voxels and without knowing what signals each particular voxel samples. So in conclusion, what does it mean to understand how the brain works? Well, understanding is achieved for a task when we have a brain computational model that performs the task and explains brain and behavioral data. How can we test brain computational models that perform real-world tasks with fMRI? We can infer brain computational models by comparing measured and predicted representations at the level of summary statistics of the representational geometry. And how can we account for the way fMRI reflects fine-grained neuronal activity to infer computational mechanisms? We can do probabilistic inference, which can rely on simulations of the population of possible measurement channels, accounting for local averaging or for more complex effects of measurement. Thank you.
Thank you, Nico. I would like to thank all the speakers. And I'm sure I can say on behalf of all the speakers that we will be happy to accommodate comments and questions throughout the meeting. Thank you very much.